Welcome to the RNA Sequencing Bootcamp, sponsored by the FGTB Council. Prior to attending the classroom session of the bootcamp, please review the following presentation and slides. This presentation will give you a primer for understanding the basics of RNA sequencing so that you'll be prepared not only for the bootcamp session, but also for designing your own RNA sequencing experiments in the future. To begin, you may be wondering what is RNA sequencing, or RNA-seq, and why investigators choose to do it. RNA-seq is a method for performing unbiased quantitative profiling of the transcriptome. By leveraging the massively parallel sequencing capacity of next-generation sequencing technology, RNA-seq allows for the large amount of nucleotide data contained in the human transcriptome to be captured in a time-efficient manner. The data generated from RNA-seq can be used for multiple purposes because RNA-seq technology allows for an investigator to look at the transcriptome of experimental or clinical samples with single-base resolution. As you can imagine, this opens the door for far more than differential expression analysis, which can be captured through older microarray methods. For example, in addition to looking at protein coding gene expression, investigators use RNA-seq for studying non-coding RNAs, novel unannotated transcripts, alternative splicing, alternative polyadenylation, RNA editing, and allele-specific expression of genetic variants, to name a few areas of RNA biology. As an investigator about to embark on an RNA-seq experiment, you'll be going through multiple steps from the wet lab to the dry lab to ensure that you generate the data you need for your project. These steps are summarized on this slide. First, you'll design your experiment or RNA collection from a clinical cohort to ensure you have enough samples to power your study and that the choices you make at each of the following steps facilitate your RNA-seq goals. Next, you'll isolate RNA from the samples you want to sequence, and either you or a core service laboratory will generate cDNA libraries that get read on a high-throughput sequencer, usually at a core facility. Once the sequencing is complete, you'll then be able to download and analyze your data if you have some experience in computer programming. If this isn't feasible, you may collaborate with a computational group or pay for computational analysis services. To give you an idea of what happens after you isolate RNA, we'll go through a general overview of the workflow. The schematic depicted here from a review by Wang et al. Illustrates the steps of how RNA is processed for RNA-seq. For the sake of simplicity, here we'll focus on long RNAs, such as messenger RNAs or mRNAs. We'll touch upon long non-coding RNAs later. Briefly, after RNA isolation, mRNAs undergo poly-A selection or ribosomal RNA depletion, and then are converted into a library of cDNA, or complementary DNA, fragments ideally greater than 100 base pairs in length. Sequencing adapters, depicted here in blue, are subsequently added to each cDNA library fragment. These adapters contain identifier barcodes that allow for multiplexing of different samples to run in the same lane. A short sequence is read from each cDNA using high-throughput sequencing technology, which we'll discuss using the Illumina platform as an example. The resulting sequencing reads are aligned with the reference genome, for example, HG19 for human samples, or MM9 for mouse samples. These mapped reads are then analyzed for differential expression of genes between control and disease or experimental conditions. There are different types of sequencers you can choose for your RNA-seq experiment. A commonly used one is the Illumina platform, which is depicted on this slide. Illumina sequencers use solid phase amplification on glass flow lanes that have a lawn of primers embedded on them. These primers anneal to the cDNA libraries that are then amplified in clusters. The amplified clusters are then read through cyclic reversible termination using fluorescently modified nucleotides that are imaged as each nucleotide is added. RNA-seq is a powerful tool for studying the transcriptome, from its regulation to different types of RNA that are differentially expressed. Knowing the types of questions you'll want to ask in your RNA samples is critical for making methodological choices that will facilitate answering those questions in your experimental design. For example, one basic question is, which RNA species do you want to study? If you want to study small RNAs such as microRNAs, you'll want to make sure your RNA isolation prep is optimized to capture small RNA species, as not all methods or kits are designed to do this. Another question to ask is if you'll be looking beyond differential expression of transcripts. If you know that you'll want to study biological functions such as alternative splicing, you'll need a read depth of at least 30 million reads per sample, but more ideally 75 million reads per sample. After your RNA-seq reads are mapped, you'll then need to choose the appropriate software program to analyze your data for your questions. Once you have answered those fundamental questions, it's important to consider the number of biological and technical replicates you need to power your experiment. Although larger sample size provide more statistical power, it's not always feasible from a cost perspective to sequence very large numbers of samples. Unfortunately, the field has not reached a consensus in terms of how to calculate adequately powered sample sizes, but there are a few studies that look at numbers of replicates needed. For example, it's recommended that you have at least 4 replicates, preferably biological replicates, per experimental group if you're looking at messenger RNAs, and 6 replicates per group for non-coding RNAs. More replicates lead to more power, whereas increased sequencing depth does not increase power for differential expression analysis of messenger RNAs once you've reached the threshold of about 20 million reads per sample, given that messenger RNAs do not require deep sequencing to quantitate. This is in contrast to non-coding RNAs, splicing events, and editing events that do require more depth for detection and differential analysis. Now that you know how you want to set up your RNA-seq experiment in terms of how many samples to use, it's time to isolate RNA from those samples. Again, the type of RNA you want to study will affect your approach in RNA isolation. If you want total RNA, Trizol can capture everything, although you'll need to make modifications in ethanol precipitation and use of carriers to ensure that you capture enough of the small RNAs if you want to study them. Alternatively, you can use commercial kits that are designed to enrich for different fractions. Regardless of the method of RNA isolation you want to use, your total RNA should have an RNA integrity number, or RIN number, greater than 7 to ensure that you're sequencing mostly high-quality, undegraded RNA. RIN numbers can be assessed on a bioanalyzer, a process that can be performed at most campuses. With a low RIN number, you may have reads with significant 3' bias, which may decrease confidence in your downstream analysis. Once you have ensured that you have high-quality RNA, you can aliquot RNA amounts according to required input, which varies according to the type of library you choose to make. Some examples, which we'll describe next, are listed on this slide. Prior to isolating RNA, you have to have an idea of what you want to study. For example, studying microRNAs or single-cell transcriptomics will require specialized library prep methods that do not cross over to other types of library preps. If you're looking at long RNAs, such as messenger RNAs, you can choose between poly-A selection, which will capture messenger RNAs and long non-coding RNAs, many of which are polyadenylated, and ribosomal depletion, which will include total RNA except for ribosomal RNAs. If you choose to pursue a ribosomal depletion method, you'll need to ensure adequate DNase treatment of your RNA and be prepared to have a slightly higher ribosomal RNA fraction in your reads. The advantage of using ribosomal depletion is that you'll capture antisense transcripts that often are not polyadenylated, as well as other non-coding species such as backspliced circular RNAs. Both poly-A and ribosomal depleted libraries must be stranded if you want to do analyses beyond protein coding differential expression analysis. If you want to capture all species of RNA in one go, you could consider TGIRT libraries, which are generated from a different reverse transcription process. More information can be found in the reference listed at the bottom of the slide. When making your sequencing libraries, there are tricks that deviate from the manufacturer's protocol to enhance the quality of your reads. Certainly, collaboration with RNA biologists who do frequent RNA-seq experiments can be very useful in learning more specific tricks. Here, we share a few things we've picked up from our colleagues. One is that if you're choosing to pay for 100 base pair reads for your experiment, you want to make sure that your libraries are actually over 100 base pairs long so that the sequencing depth you paid for is not wasted. One way to do this would be to decrease the fragmentation time by 2 minutes in the library generation protocol. This usually ensures most fragments are at least 150 base pairs long. Another concern in RNA-seq is potential sources of bias at the library preparation stage. A source of bias is certain runs of sequences that get amplified more. For example, GC-rich and GC-poor transcripts may be underrepresented. And reduction of PCR cycles for library amplification reduces this bias effect. Even differences in barcode sequences used for multiplexing can lead to differences in adapter ligation and library prep, as well as amplification biases. So barcodes chosen for your multiplexing will need to be considered carefully. For more information, references are listed on this slide. Now that your libraries are made, you have some choices in terms of how you want to sequence them. For example, different analytical goals will require different depths of sequencing, from lower reads for messenger RNA differential expression analysis, to very deep sequencing for de novo assembly of transcripts, which is important if you're, for example, trying to annotate novel non-coding RNAs. You can also choose whether you want paired reads, which will give you better resolution at each base and more confidence in calling events such as alternative splicing. Likewise, longer reads will give you better coverage of a transcript, which will be needed for more sophisticated analyses. With all of these choices, we're sometimes limited by cost. The website listed on this slide provides a price comparison among different sequencing cores and companies around the country. One type of sequencer that's frequently used is the Illumina HiSeq 2500, which contains 2 lanes per flow cell for a rapid run, 8 for a high output run. With more than 1 lane available to you, you can multiplex libraries to run multiple samples down 1 lane. With multiplexing, you then have the freedom to pool as many libraries with different barcodes on a given lane. For the HiSeq 2500 rapid run, you want to avoid batch effects by mixing your samples as much as possible. For example, if you have an experiment consisting of 8 samples with 4 replicates per group, running all 8 samples on each lane gives you the same depth as splitting them up so that all of one group is in one lane versus the other group in the other lane. As you can see, you may be introducing a batch effect if you were to choose to pool your libraries in the approach shown on the left. To minimize batch effect, you want to pool your libraries in the approach used on the right to decrease bias from unpredicted differences in each lane's run. After your sequencing run is complete, it's time to process and analyze the data. Here's an overview of how data is processed from the sequencer to a text output of differential expression analysis. First, the facility that performs your sequencing will release to you the raw data in the form of FASTQ files, which also usually contain some basic quality control metrics, such as the percentage of read nucleotides meeting a quality Q30 score, which has a 99.9% base calling accuracy. Because your samples were multiplexed with adapters and barcodes, they need to be trimmed so that the adapters do not cause significant mismatches in alignment to the reference genome. Aligning reads to the reference genome is the way one identifies to which gene a transcript read belongs. After alignment, you can do further library QC to assess whether you have good library complexity of uniquely mapped reads. A high duplicate read rate for messenger RNAs means that many of those reads are artifact from amplification clones rather than true reads of transcript abundance. This is because the RNA fragmentation process for library generation would generate many random breaks in transcript copies of the same gene, and thus unique fragments, rather than fragments of the exact same sequence. If library complexity is poor, you may need to consider redoing and troubleshooting your libraries. Additional library QC includes looking at 3' bias to assess whether your sequence transcripts are intact or degraded. Once you're happy with the quality of your libraries, you'll then proceed with quantifying transcript abundance and then testing for differential expression between sample groups. Here are examples of commonly used adapter trimming programs. With the adapters trimmed off your FASTA files, you must then align your reads to the relevant genome. This concept is reviewed in the next slide. In the case of our bootcamp, we'll be using human data and thus aligning to the human genome. 2 commonly used programs are listed here. To review what we mean by aligning to the genome, let's say one RNA-seq read is represented by the blue puzzle piece. This piece must be matched to the corresponding portion of the genome from which the original RNA was transcribed. Identifying the position of the genome to which the puzzle piece aligns allows for identification or mapping of the relevant gene or transcript, as many genes and transcripts are already annotated. Mapping of an aligned read to an annotated gene or non-coding RNA occurs in the analytical phase. You'll find that there are many publicly available annotation files from which to choose, and they can include protein coding genes as well as various species of non-coding RNAs. Commonly used annotation files are those from Ensembl, GenCode, UCSC, and RefSeq. After aligning your data, you are not quite ready to begin your analysis. Prior to analyzing your data, you need to ensure that the quality of your libraries is acceptable. For long RNAs, you want complex libraries that have a low duplicate read rate. What do we mean by duplicate reads? Think back to what we said about fragmenting RNA for generating libraries. Fragmentation is a random process, and it'd be extremely rare for one transcript copy to be fragmented into the exact same greater than 150 base pair pieces as a different copy of that same transcript. Therefore, a high duplicate read rate, which is commonly defined as greater than 30%, although there's no consensus on what's acceptable, is indicative of PCR clones in the library amplification step that get preferentially amplified, rather than necessarily truly higher transcriptomic expression. Another quality measure we examine is 3' bias, which is indicative of RNA degradation. This may affect the confidence you have in your data. The graphs here represent read coverage along the length of transcripts. Even coverage is represented in the graph on the left, where you can see that the 5' and 3' ends are represented fairly equally. The graph on the right is an example of having more reads on the 3' end, which may not be suitable for analysis. Once you're happy with your libraries, you'll proceed to analytical programs to generate the data you want for your papers and grants. This slide lists the two most commonly used differential expression programs, DESeq2 and CuffDiff2, as well as new, up-and-coming programs Callisto and Sailfish. Once you or a computational research specialist have run your processed RNA-seq files through your program of choice, you'll receive a dataset output from which you can run further analyses to find interesting targets. To get you comfortable with the output you might see, we'll show you examples from DESeq2, which will be used for the classroom portion of the bootcamp, and CuffDiff2. There's no right or wrong program to use. Your choices may be based more on the statistical methods you believe in more. Here's an example output from DESeq2. The base mean is the mean of normalized read counts for an annotated transcript. That transcript has a fold change in expression between experimental groups. And here the fold change is expressed on the log2 scale and is called the log2 fold change. IFC-SE stands for log fold change standard error. You also receive the output of the Wald test statistic, the p-value, and a p-value adjusted for a false detection rate, or FDR. We'll touch more on FDR in a bit. This is an example of output from CuffDiff2. You can see the control group and the variable group, as well as the corresponding FPKM values of each group. What do we mean by FPKM? FPKM is fragments per kilobase of transcript per million mapped reads. In essence, it's a calculation to normalize the number of reads by the length of the transcript, as a longer transcript may have more total read fragments mapped to it by virtue of it being long. FDR adjusted p-value, or q-value, is more rigorous statistically than a p-value. For IDO1, the first gene in the list, there's a 0.05% chance among all expressed genes, which can be in the thousands, that IDO1's called differential expression pattern would be a false positive, whereas the q-value implies that 0.25% of genes with q-values less than 0.0025, a small number of genes, will be false positives. Because q-values are more rigorous, they're often the values reported in manuscripts. We hope that this presentation has given you background for what you need to know for the classroom portion of our bootcamp, where we'll have you perform additional analyses, such as gene ontology analysis, on a real RNA-seq dataset processed for differential messenger RNA expression. See you at the bootcamp.